All right, welcome everybody to this lecture on cellular respiration. I want to start by just looking at this crazy looking picture here that shows a complex series of metabolic pathways to give you sort of an appreciation of how complex biochemistry can be. We're not going to, of course, memorize this kind of chaos here, but I just want to show you to give you appreciation of how complex this type of thing is. What we're going to do, we're going to talk about cellular respiration today. And cellular respiration, what we're going to do in cellular respiration is we're going to take, in this series of biochemical reactions, we're going to take the food that we eat, and in this case, we're going to start talking about glucose because it allows you to sort of go through all the steps. We're going to take glucose and we're going to put it into the machinery of the cell, if you will. And what we get from that is we get energy out of that. So what we're going to do is we're going to take the energy that's in glucose and we're going to transfer the energy to another form of chemical energy. And that is going to primarily be, as you will see, ATP. Because most of the cell functions that go on in biochemistry, at least, rely on the energy source of ATP. Before we do that, as we start into that, one of the things you need to understand is we have another, I mentioned before, there's several different types of chemical reactions that happen. This is what we call a redox reaction, and that is for oxidation and reduction. So in this type of reaction, what ends up happening is one molecule or several molecules are going to become oxidized, and then other molecules are going to be reduced. Okay, so in this particular set of reactions, what's going to happen is glucose is going to be oxidized. And what happens when you have oxidation is you're going to lose hydrogens from that molecule and therefore lose the electrons that go with it in something that is going to be, if you have an oxidation reaction going on, it's always tied to another reaction called a reduction action. So in this particular case, oxygen is going to be reduced, and reduced means it's going to be gaining electrons. So you can think about electrons, keep this in mind that they're negatively charged. An electron is negatively charged. If you gain something negative like a credit card bill, if you think about that, then you have reduction going on. It's reduced. Okay, so that's how I like to remember it. And the summary of this here is designed to sort of just show you that we're going to break this down into 4 steps. Different people do this different ways. I'm going to break it down into 4 steps. Glycolysis, there'll be one in here that we will call the PDC, as you will see, the Krebs cycle, and the electron transport chain. And so as we go through these set of reactions, you notice you get a little bit of ATP there, a little bit of ATP there, but this is where you're going to get the most ATP, something on the order of 34 to 36. Different textbooks differ on this number, so I'm not going to worry about being exact. There's some biochemical reasons why that is, but certainly most of it comes out in this last step, the electron transport chain. And what I want you to focus on, I'm going to walk you through all the steps, and I'm going to show you what happens, for example, in glycolysis and PDC and so forth. But what I want you to remember is I want you to remember what the inputs are, what goes into that reaction, what comes out of that step of reactions like in glycolysis, and what the main goal is, what was accomplished, what's important about glycolysis, what's important about the PDC, Krebs cycle, and electron transport chain. And then I want you to know overall what's the main goal of cellular respiration, what's accomplished, why is it done, and that is to make ATP. Okay? And the reason we need that's like if you're going to move your muscles, if you're going to think, all these processes require energy to do that, to maintain homeostasis, and that energy comes in the form of ATP. Okay? So we'll start with glycolysis first. And you notice that when we start with glycolysis, we start off with glucose. And you'll notice there's a series of reactions, and with each reaction there's an enzyme. So for example, in the first step, glucose gets changed into glucose 6-phosphate by actually using a little bit of ATP and transferring that phosphate using an enzyme hexokinase, okay, to make glucose 6-phosphate. Then glucose 6-phosphate becomes fructose 6-phosphate using the enzyme phosphoglucose isomerase, okay? And that continues on, and there are enzymes for each step. And then at the end, when we're done going through this series of reactions you see here, what we end up with at the end is pyruvate or pyruvic acid. So you can see that right down there on the bottom. You do not need to know for Bio 1 in this course each enzyme, each intermediate molecule, and that's why I was saying on that summary chart back there, we want to focus on what goes in and what comes out. All right, so there's your pyruvate. You'll notice also we're putting in ATP, but we get ATP there and we get ATP out there. So overall, what we get about 2 ATP molecules from this set of reactions, okay? We also are going to get, I'll explain this here in a second, this NAD plus and NADH, okay? And that's going to be an important molecule later on, NADH, so we're going to talk about that, okay? So I went too far. All right, so when we talk about NAD plus and NADH, this is what NAD plus looks like. This is what NADH looks like. And if you notice, they're very identical looking molecules. And the difference lies right up here. So if you look at that versus that, there's a little bit of a difference here, that extra hydrogen there. And what I want you to think about is that what happens is NAD plus, we're going to transfer the energy along with the energy in a high energy electron and a hydrogen from NAD plus to NADH. So NAD plus becomes NADH once it grabs a hold of a high energy electron. So it's kind of like, it's similar to when we have ADP and ATP and ATP. These are basically one molecule turns into the other, and this is the high energy state. That's what happens in this case here. So NAD plus becomes NADH, and it's holding on to, at that point, a high energy electron. And that's very important in glycolysis because we're going to use this NADH and its high energy electron in the final step. Okay? All right, so once again, just so you're understanding what's going on here, we're taking glucose, and the little arrows show you, okay, these are steps, and that's a step, and that's a step. So instead of showing you all the enzymes here, what they've done is they've just sort of shown you what you get out of it. So here I take my glucose molecule, I use a little energy putting into that, that ends up splitting into 2 molecules, and when I'm all done, what I get is I get my 2 pyruvic acids or pyruvate. You can call those, either one is fine with me in this particular class. I get ATP. So you notice I get 4 ATP, but I had to use up 2 there. So I still get 2 ATP out of it. And I get this NADH molecule, which is again a high energy electron storing molecule that I'm going to use later on. Okay, so that's glycolysis. The next step is what we call the PDC, or the pyruvate dehydrogenase complex. And what's going to happen in this case is we're going to take that pyruvic acid. Okay, there's my pyruvic acid. We're going to break off. There's going to be an enzyme to break off one of these carbons here. And that's where my CO2, or one of the places I'm going to get CO2 coming out. So as you should probably know by now, cellular respiration, as we use up energy and transfer it, one of the waste products that we produce that is in our breath when we breathe out is CO2, which is a waste product. It's something that comes out of this series of reactions. So, for example, if you were to exercise, the more you exercise and the more you run, the more your body needs to use up energy to perform that exercise. Your cellular respiration rate is going to increase. You're going to produce more CO2. So once I break off that carbon dioxide, my pyruvic acid then becomes acetic acid. And then what happens is my acetic acid, I'm going to attach it to this molecule called coenzyme A. And I'm going to once again use some of the energy that's in that to take NAD+, transfer a high energy electron to it to make NADH. And once I hook that coenzyme A to my acetic acid, I get as my output, the thing that comes out of the PDC, or the pyruvate dehydrogenase complex, is this acetyl coenzyme A. And that is going to be the product that comes out that goes into my next step. So as you can see here, as we get ready to go into the Krebs cycle, what ends up happening is there's my coenzyme A. So there's my pyruvate. Okay, there's my coenzyme A. And my coenzyme A is going to be recycled. So my coenzyme A gets hooked to that and then gets taken back off. That's used to transport really this into the third step, which is called the Krebs cycle. So the Krebs cycle, also called the citric acid cycle. So you see different terms for that. Okay, citric acid cycle. What's going to happen, that's what they have here, citric acid cycle. And so what I'm going to do is I'm going to take that 2-carbon molecule, I'm going to hook that to a 4-carbon molecule, and I'll be back to a 6-carbon molecule. So what happens as you go through, once again, there's an enzyme that runs each of these reactions, and you don't need to know the names of these intermediate molecules for Bio 1. Okay, in bio, majors bio would make you do that, make you learn the enzymes in biochemistry. But for this particular class, for non-majors, general science class, it's okay to know what goes in and what comes out. And so, once again, you've seen this reaction before. Here's my NAD+. I'm going to make NADH. Out of that, I'm going to transfer the energy from this to NAD+ to get NADH. There's more NADH. There's more NADH. This one here is very similar. This is FAD becoming FADH2. What you can think about is that NAD plus is kind of like FAD, and NADH is kind of like FADH2. But this is a lower energy intermediate. Okay, so although they're similar in what they're doing, you can just think about they're similar, but this one has a lower energy state. And once again, also you notice that in the Krebs cycle, I get a little bit of ATP right there coming out, and I'm also releasing the waste product CO2. Okay, so at this point, we've produced something like about 4 ATP, which is not very many. In the final step, what we're going to do is, this is called the electron transport chain, also called the ETC, so the electron transport chain. And you might be able to guess at this point, what does the electron transport chain do? It transports electrons. So this is going to happen across the inner membrane of the mitochondria. So remember, in a cell, you have a cell with a nucleus, for example, and then you have other organelles in here, some of which can be these mitochondria. And remember, the mitochondria has 2 membranes. It has one on the outside, and it has an inner one here. And that's what you see here. This is the inner membrane right there. And so what we're going to do is, on the inside of the mitochondria, which is called the matrix in here, which is right down here, what we're going to do is our NADH, which again is holding on to these high energy electrons, we're going to transfer the energy to these proteins. Okay, and these are the proteins that are in the transport chain, the electron transport chain. So we're going to transfer the energy and the electron, and we're going to, as we do that, as we move the electron down from step to step, what we're going to do is we're going to transfer the energy and use that to pump hydrogen ions across through that protein into this space, the inner mitochondrial membrane, between the outer and the inner mitochondrial membrane, in that little space there. So that's what we're using the energy for. So we're pumping. These are all essentially going to be pumps as we think about if you have like a stairwell, okay, and you're rolling a ball down the stairs, okay, it's releasing energy as it goes. Okay, instead of the energy just escaping, which some of it does, once again, because the second law of thermodynamics, you're always losing some of that energy as heat. Instead of just losing the energy to the ball just falling down, we're going to use that energy to pump hydrogen ions across into this space. Okay, now at the end, as that electron travels down, it reaches the final end point here, and the electron then gets stuck right here, and you need somewhere to put that electron or this whole chain backs up. And that's where oxygen comes in. And you don't have to worry about this 1/2 O2. They do that so this equation will balance out, but you can just call it O2. That's fine with me. So what happens here is we use oxygen, and oxygen ends up being what we call the final electron acceptor. All right. And so what happens is that electron with the oxygen ends up becoming water. So you produce water in this reaction. And this is why you need oxygen. And this is why aerobic exercise is so necessary for generating large amounts of ATP, as we'll see here in a second. So that's a final electron acceptor. Now, once again, we've built up this big giant sort of waterfall of hydrogen ions, if you will. So we have a whole bunch of hydrogen ions all pumped out here. Now, since we have them in such high concentration out here, they're going to flow from high concentration to low. So they're going to go through this pump here, or actually this protein, and this protein is called ATP synthase. And what's going to happen is, as they flow through the protein, we're going to use the energy as these hydrogen ions flow through here. We're going to use the energy to take ADP and transfer that energy from the hydrogen ions flowing through to make ATP. So I don't know if I said that correctly, just to make sure. We're going to use ADP plus inorganic phosphate to make ATP. So this is a concentration gradient. You could think of it sort of like if this was a waterfall. If you put a whole bunch of water up on a hill and it was flowing down the hill, the energy as the water flows down the hill could be used to turn, a wheel or something like that. But in this particular case, we have a concentration gradient, and as it goes through, it releases that energy, and this protein ATP synthase, which you see here in a little more detail. Your ATP synthase takes the hydrogen ions, and as it goes through, this thing's like a little machine here, this protein, and it's used to transfer the energy from the flow of hydrogens to make ATP at the end. So once again, this is a summary just to show you sort of all the steps together. We started with glycolysis, then the PDC, then the Krebs cycle, then the electron transport chain. And what you want to keep in mind is think about what went into each reaction, each step of reaction, what comes out of each reaction, and what was the main goal. So for example, in glycolysis and the PDC and the Krebs cycle, what we really accomplished that's so useful is, although we make some ATP there, and here as well, should add, although we make some ATP, what's really important is that we're making this NADH. We're transferring the energy from the food molecules, we break it down to make NADH, as well as a little bit of FADH2 as well. And when you look at this, what happens in glycolysis, we make a little bit of ATP. In the PDC, we give off some CO2. We make some NADH, that's important. In the Krebs cycle, we make we take in acetyl coenzyme NAD plus and FAD. This is what we get out of it. We get ATP, NADH, and FADH2 as our main important goals from that. But it's really the electron transport chain where we use up oxygen as our final electron acceptor, which ends up giving us water, where we make most of the ATP, somewhere on the order of 34 to 36 ATP from the whole process.